Dear audience, I am Alexandra Korostyshevkaya from International Tomography Center of the Siberian branch of the Russian Academy of Sciences, Novosibirsk, Russian Federation. I will talk about our recent publication in Journal of Magnetic Resonance Imaging titled Direct Comparison between Apparent Diffusion Coefficient and Macromolecular Proton Fraction as Quantitative Biomarkers of the Human Fetal Brain Maturation. I'll present only highlights of the paper. For further details, please refer to the paper itself. Fetal brain development is a complex process involving rapid changes in tissue morphology and microstructure. Various quantitative MRI methods were used over the past decade to characterize tissue changes during the fetal brain maturation. Among such methods, diffusion-weighted imaging (DVI) gained substantial popularity due to wide availability, fast acquisition, and sensitivity to changes associated with gestational age. Fast acquisition is especially important in fetal studies because of fetal motion. The apparent diffusion coefficient (ADC) demonstrated high sensitivity to changes in microstructural tissue organization in the developing brain. Recently, a new quantitative neuroimaging method, fast macromolecular proton fraction (MPF) mapping, has been adopted to the fetal brain imaging. MPF is a biophysical parameter defined with the magnetization transfer model, which describes the amount of macromolecular protons in tissues. MPF has found to be closely associated with the myelin content in brain tissues. Several earlier studies confirmed strong correlations between MPF and histology in animal models. This method allows MPF mapping using a single magnetization transfer image and therefore provides sufficient speed for many clinical applications. The fast MPF mapping method showed high sensitivity to the early stages of myelin development in the fetal brain. You can find more details in our recent paper in American Journal of Neuroradiology. As both ADC and MPF can reliably detect changes in the fetal brain during its maturation and allow fast scanning, the goal of our study is to compare these parameters directly. This was a cross-sectional study performed in a clinical diagnostic radiology setting using 1.5 Tesla scanner. We performed a quantitative MRI protocol as an addition to the standard clinical protocol with the extra scan time of about 5-7 minutes. We recruited a total of 55 pregnant female patients referred for various clinical indications. For this study, we included only fetuses over 20 weeks of gestational age without serious brain abnormalities found on clinical images. We have also excluded cases with severe motion artifacts. The final study population included 42 fetuses. For subsequent analysis, we grouped the fetuses according to the pregnancy trimesters, and there were 21 fetuses in each of the second and third trimester groups. As I mentioned, the MRI protocol was implemented for a standard clinical scanner, 1.5 Tesla Philips Achiever. I'd like to emphasize that we used standard sequence without any scanner software modifications. Diffusion-weighted imaging was performed using a standard single-shot echo-planar sequence with the 5B values. The ADC maps were reconstructed using scanner software. You can see example diffusion-weighted images and a resulting ADC map in this slide. Fast MPF mapping was implemented according to the single-point method described earlier. You can find the details of protocol implementation in the discussed paper and in our earlier publication in the American Journal of Natal Radiology. In brief, the method is based on the nonlinear fit of the magnetization transfer model, and it uses 4 source images to reconstruct an MPF map. The images were obtained with a spoiled gradient echo sequence and provide different types of contrast weightings. Specifically, there is an image with a magnetization transfer saturation, a reference image with the same parameters and without saturation, T1 weighted image, and the proton density weighted image. You can see also a resulting MPF map reconstructed from these images using custom software. In this slide, you can see more quantitative maps of the fetal brain and the scheme of image analysis. There are example ADC and MPF maps of 2 fetuses with gestational ages of 20 and 35 weeks. For quantitative analysis, MPF and ADC values were manually measured in the regions of interest placed in the following brain structures: medulla, cerebellum, pons, thalamus, temporal, frontal, and occipital white matter. 2 observers performed the measurements and the inter-observer agreement was generally good. Statistical analysis included several procedures. To compare ADC and MPF values between brain structures and pregnancy trimesters, we used mixed repeated measures, analysis, and of variance. We also tested persons' correlations between ADC, MPF, and gestational age for each structure and for the entire dataset. Finally, we used a Hartelling-Williams test to compare strength of correlations. Now, let's take a look at the results. This slide demonstrates the summary statistics of our observations. Analysis of variance revealed highly significant effects of the brain structure and the pregnancy trimester on both ADC and MPF. Their interaction was also significant. The plots show the results of pairwise comparisons for ADC left and MPF right. Asterisks indicate significant differences between the second and the third trimesters of the same structure. Shared letters mean the absence of significant differences between structures within the same trimester. As we can see, MPF increases from the second to third trimester in the brainstem structures, pons, and medulla, as well as in the cerebellum and thalamus. These structures are known to have prenatal myelination onset. ADC significantly decreases only in the cerebellum and thalamus. At the same time, no distinctions between trimesters were observed in the cerebral hemispheres. It is also known that myelin in cerebral white matter develops during the postnatal period. Now, let's take a look at the distinctions between structures. We can appreciate that the medulla and pons show high ADC and lower MPF. We can also see that the spread between the structures increases in the third trimester. Finally, if we compare behavior of ADC and MPF, we can appreciate that MPF reveals a greater number of significant differences between the structures, as indicated by more letters in the right plot. The next slide summarizes correlations between ADC, MPF, and gestational age in the anatomic structures of the fetal brain. Significant correlations between all variables were found in the pons, medulla, cerebellum, and thalamus. For frontal, occipital, and temporal white matter regions, we didn't find significant correlations. Corresponding plots are not shown here and can be found in the article. You can see that correlations with gestational age are remarkably strong for MPF, with their correlation coefficients exceeding 0.8. At the same time, correlations between ADC and gestational age are of moderate strength. Finally, it is important to point out a substantial similarity in overall trends displayed MPF and ADC. This plot demonstrates correlation between MPF and ADC averaged across brain structures. Although correlations between these parameters in separate brain structures are not very strong, as we have seen in the previous slide. Correlations across structure average data appear much stronger. Furthermore, the strength of correlation increases from the second to the third trimester. These observations suggest that myelination is the main driving force for changes in both MPF and ADC in the fetal brain structures. In summary, our results show that MPF and ADC demonstrated substantial qualitative similarity in the spatial-temporal patterns of the prenatal brain development. Particularly, both ADC and MPF showed an apparent pattern of divergence between different brain structures in the third trimester compared to the second one. Both parameters also demonstrated significant correlations with gestational age in the anatomic structures with known prenatal onset of myelination. At the quantitative level, MPF appeared more sensitive than ADC to the prenatal myelin development. Finally, I want to emphasize that ADC and MPF maps can be obtained in routine clinical settings and may provide complementary quantitative information about normal or abnormal brain fetal development. At the end, I would like to acknowledge grant support from the Ministry of Education and Science of the Russian Federation, Russian Science Foundation, Federal Agency of Scientific Organizations of the Russian Federation, and the United States National Institutes of Health.